Lactobacillus bulgaricus is a species under the lactic acid bacteria, which is used in the production of yogurt and other fermented dairy products.
Lactobacillus bulgaricus is classified as follows. It falls under kingdom Monera, phylum Firmicutes, class Bacilli, order Lactobacillus, family Lactobacillusi, genus Lactobacillus, and species bulgaricus, hence the scientific name Lactobacillus bulgaricus.
My name is Rashida.
My name is Kavin.
Daniel, join us as we explore more about lactic acid bacteria.
Some of the key features of Lactobacillus bulgaricus. It's generally microscopic in nature, where its width ranges between 0.5 to 1 micrometer. The length ranging between 2 micrometers and 2 micrometers. It falls under class Bacilli, therefore it's rod-shaped. It's also non-motile, since it lacks flagella. It's also non-spore producing. So Bacillus bulgaricus is a facultative anaerobe which metabolizes in presence and absence of oxygen. So in this, Lactobacillus bulgaricus breaks down lactose into lactic acid, which is crucial in yogurt production. It is also thermophilic in nature, where it thrives between temperatures of 40 to 45 degrees Celsius, and also it's probiotic in nature, where it contributes to the gut health if taken in adequate amounts.
Everyone, today we are going to look at the optimum growth conditions and requirements of Lactobacillus bulgaricus and how these conditions relate to its presence in food. Lactobacillus bulgaricus is a type of lactic acid bacteria commonly used in the production of yogurt and other fermented dairy products. This bacteria thrives under specific conditions that promote its growth and activity. Here are the optimum growth conditions and requirements for Lactobacillus bulgaricus. There is temperature. Lactobacillus bulgaricus grows best at temperatures between 37 to 43 degrees Celsius. This temperature range is ideal for its metabolic processes and reproduction. Nutrients. Lactobacillus bulgaricus requires a source of carbohydrates such as lactose in milk to fuel its growth. It also benefits from the presence of vitamins and minerals for optimum metabolism. pH. Lactobacillus bulgaricus prefers a slightly acidic environment with a pH range of 4.0 to 5.4. This acidic pH helps inhibit the growth of harmful bacteria while promoting the growth of Lactobacillus bulgaricus. Oxygen. Lactobacillus bulgaricus is a facultative anaerobe, meaning it can grow in both the presence and absence of oxygen. However, it typically thrives in anaerobic conditions. Then time. Lactobacillus bulgaricus requires sufficient time to ferment and produce the desired products such as lactic acid and flavor compounds. In relation to its presence in food, Lactobacillus bulgaricus plays a crucial role in the fermentation process of dairy products like yogurt. During fermentation, Lactobacillus bulgaricus consumes lactose in milk and produces lactic acid, which helps carry the milk proteins and gives yogurt its characteristic tangy flavor and texture. So the optimum growth conditions mentioned above are carefully controlled during the production of yogurt to ensure the growth of Lactobacillus bulgaricus and the desired fermentation process. By providing these ideal conditions, food producers can promote the growth of Lactobacillus bulgaricus and produce high-quality fermented dairy products like yogurt.
Are the highlights on the significances and contributions of Lactobacillus bulgaricus in the food processing industry with the practical application of Lactobacillus bulgaricus in the food microbiology. Lactobacillus bulgaricus is crucial in food for its role in fermentation, particularly in yogurt production, where it converts lactose into lactic acid, contributing to yogurt's tangy flavor and thick texture. Lactobacillus bulgaricus is used in the production of probiotic foods because it is considered a probiotic bacteria, meaning it provides health benefits like improving digestion and boosting the immune system. It enhances the shelf life of yogurt by creating an acidic environment that inhibits the growth of harmful bacteria, thereby preserving the freshness and the quality of the product. Lactobacillus bulgaricus helps in the reduction of lactose intolerance, where it produces lactase, an enzyme that breaks down lactose into lactic acid, making it easier for people with this issue to digest dairy products. Lactobacillus, its contribution to food processes extends beyond yogurt to various fermented dairy products such as buttermilk, certain types of cheese. Lactobacillus bulgaricus is crucial in flavor and aroma development, where it develops the characteristic flavor and the aroma of fermented dairy products through the production of aromatic compounds like the acetaldehyde and acetone, and thus contributing to the sensory properties. I hope we have highlighted the significance and the practical application of Lactobacillus in the food industry.
Hi everyone, this is Daniel together with
Joseph,
and together we shall be discussing the isolation and purification of Lactobacillus bulgaricus, a key probiotic bacteria used in the dairy industry, particularly in the yogurt production.
Lactobacillus bulgaricus is a gram-positive, rod-shaped bacterium. It plays a crucial role in the fermentation of dairy products. Benefits include aiding digestion and suppressing harmful bacteria.
So the objectives of this video are to isolate Lactobacillus bulgaricus from yogurt sample, then to purify and identify the isolated strains through morphological and biochemical methods.
So the materials required, we are going to use a yogurt sample, which is this one. We need test tubes containing rigorous reagent. This is a diluent, so these ones contain rigorous reagent. So we are going to use MRS reagent, MRS agar plates. We are also going to need a microscope, an incubator set at 45 degrees Celsius.
For collection for our experiment, we shall begin with commercially available or homemade yogurt known to contain live cultures. Ensure that the sample is fresh and properly stored to maintain the viability of the bacteria.
So now on isolation and preparation, we are going to use serial dilution technique. So serial dilution involves decreasing the concentration of microorganisms in a sample through a series of sequential dilutions. The samples contain too many cells to be counted, and thus to obtain plates that are not hopelessly overgrown with colonies, dilution becomes necessary. So we are going to get to dilute 1 gram of yogurt in 9 ml of rigorous reagent to create a 10 power neg1 dilution. Then that 10 power neg1 dilution, we are going to pick 1 ml and then we dilute it in 9 milligrams of another rigorous reagent, and this procedure is going to be repeated until we get a 10 power neg6 dilution.
Okay, after diluting our samples, we shall plate from the appropriate dilutions onto MRS agar plates. Spread evenly on the plate using the sterile spreader. Then after, incubate at a temperature of 45 degrees Celsius for 24 to 48 hours under anaerobic conditions.
Then for an identification, after incubation, we are going to select the distinct colonies with characteristic morphology. So the colonies are going to be small, white, and saturated with a smooth surface.
Then for the purification, we shall strip the selected colonies onto the fresh new MRS agar plates to obtain pure cultures. Incubate as before and verify the purity by restaining and testing.
Okay, the storage and maintenance, we are going to store pure cultures in MRS broth supplemented with a 15% glycerol at 80 degrees Celsius, and then maintain cultures by subculturing every 2 weeks.
So we measure 1 ml of yogurt, and as you can see, we shall add it to 9 ml of diluent. So now we have 10 power negative 1 of solution. Now we repeat this process until 10 power negative 6. Always remember to label your test tubes. We carry out plating. We first pour melted agar onto the plates and leave it to solidify. After the agar was solidified, we measure 1 ml and add it to the plates and then incubate for 24 hours at 45 degrees Celsius. So after incubation, you will see small white colonies. So now we shall carry out purification. Make sure to label your agar plates and divide it into 4 quadrants. So we shall pick a colony and then spray it on another freshly prepared agar plate. We shall then incubate for 24 hours at 45 degrees, and these are the results after incubation.